Hi, I'm Professor Johnson, and in this video we are going to be talking about enzymes. So enzymes, you recall, are usually proteins. There are exceptions to this rule. For example, there are certain RNA molecules that can act as an enzyme, and we call these ribozymes. A good example of this will be the ribosome. It's very important for protein synthesis. But for the most part, you can think of enzymes as being protein catalysts. So these are things that don't necessarily favor one direction in the reaction, but it speeds up the rate at the reaction that it goes. So it can increase the rate of the forward reaction, and it can increase the rate of the reverse reaction as well. So enzymes have some important features. One of these important features is the active site. So the active site is where the substrate is going to bind. And what is a substrate? Well, a substrate is the reactant molecule that's going to occur. We call it a substrate when it occurs in enzyme catalyzed reactions. Now, enzymes can't function on their own. They'll sometimes need small molecules called coenzymes or other types of cofactors such as ions that will allow them to run. And when those are present, you have the holoenzyme, meaning the entire enzyme that allows the reaction to occur. Now, enzyme regulation is important. You can regulate enzymes by having them present in the cell or not having them present in the cell. But if you want the cell to be able to react quickly, you want to have the enzymes there, and then you want to either activate them or deactivate them. And so enzymes can be activated by activators, or they can be inhibited by inhibitors. Now, enzymes are going to participate in biochemical pathways, and these biochemical pathways then are going to be regulated by feedback regulation. This can be either inhibition, which is what we are going to focus on more, but it can also be regulated by activation of the enzyme as well. So remember, proteins are enzymes. That means that the enzymes can be denatured. And when they're denatured, they're unfolded, and so they're not going to be able to act like they normally will. Thus, they're going to be sensitive to all the different things that can denature proteins, such as changing the pH of a reaction or changing the temperature of a reaction. So notice the graphs here. So the graphs, you can see that as you go to a certain pH, the rate of the reaction goes up until you have what is known as the optimal pH, and then it's going to start going down again. This is the pH at which the protein is properly folded to carry out its enzymatic function. And you can see the same thing happens for temperature. Right here, the protein isn't folded accurately, and it's moving slow because the reaction isn't going fast because it's a cold temperature. Then you reach the optimal temperature, and then after that you start going too high in temperature and the protein denatures, and then you lose the enzymatic activity. Now, one thing that needs to be focused on with this is, sure, many proteins will operate at optimal temperature of 37 degrees Celsius, and many of them will operate at an optimum pH of 7. But it's going to depend on the protein. Some proteins are going to function better at higher or lower temperatures. Oftentimes it depends on the organism they are in or the particular place in the organism they are in. And this is true for pH as well. And especially true for pH when we're looking at different places in the body. So here you can see that different enzymes in humans have different optimal pHs depending on where in the organism they're present. So pepsin, for example, is going to be the enzyme that helps break down proteins in our stomach. So it's going to be operating at an optimal pH that is closer to the gastric juices, so it's at a low pH. Salivary amylase, on the other hand, is the enzyme that breaks down carbohydrates in our saliva. Our saliva is mostly neutral, so salivary amylase is going to need to operate at a neutral pH to be the most efficient at what it does, and that's what we find. And then pancreatic lipase, so the pancreas, the juices in there are actually more basic than they are acidic. So they actually are going to operate at a higher pH than that. And that's going to be true for every enzyme. It's going to depend on where the enzyme is actually functioning that will determine how optimal what pH it'll optimally work at. All right, so oftentimes you hear of enzymes binding to substrate in a lock and key model, where the substrate is the key and the enzyme is the lock, and it fits in there just perfectly, it turns, and you have the reaction occur. That is not actually a perfect analogy. A better model that we now use is the idea of the induced fit model. So the induced fit model indicates that the active site where the substrate will binding isn't just a perfect fit. It's not like a puzzle piece fitting into it, but it's actually going to be shaped in such a way that it's not the perfect fit. But when the enzyme binds, you then have a change in the conformation of the protein such that it fits better. And it's actually going to fit in such a way that it's going to make it optimal for the reaction to actually occur. And that's not really depicted in here, but how it's folded, it's going to be folded sort of like in a conformation that allows the protein to react efficiently. All right, so sometimes enzymes can't operate on their own, but they'll actually need something to assist them. And this is where things such as coenzymes and cofactors come into play. So coenzymes are a specific type of cofactor that involves being a small organic molecule that binds to the enzyme and allows it to react. So often coenzymes are vitamins that you may have heard of. So riboflavin, for example, can be manufactured into many of the coenzymes that we use. So cofactors, on the other hand, are a broader category. Sometimes cofactors, or some cofactors could include magnesium ions or calcium ions or other things that are going to bind to the enzyme and help it react. So how coenzymes work is they're going to bind to the substrate, giving it a place for the reaction to occur. And now you can see the substrate came in, and then that provided a place for the reaction to actually occur. So coenzymes are things that help complete the enzyme so that the enzyme can react. All right, so sometimes molecules are going to be regulated, and they can be regulated by inhibition or activation. So inhibiting it stops the reaction from occur, activating it helps promote the reaction to occur. You can divide inhibition into how it actually binds the molecule. So it can be something that binds to the active site, or it can be someplace that binds elsewhere, known as the an allosteric site that prevents the reaction from occurring. First, we'll look at competitive inhibition. So with competitive inhibition, you have your inhibitor that's actually going to bind to the active site. So let's take a look at that occurring. So your inhibitor binds to this active site, and it prevents the substrate then from binding because it has something here. So it's competitive because both the substrate and the inhibitor are competing for the same site. Now, something that you need to keep in mind when we talk about competitive inhibition and non-competitive and allosteric inhibition is that these inhibitors and these substrate, they can bind and they can unbind. So it's in an equilibrium where sometimes you have the enzyme bound. That's what this EI here stands for. And sometimes the enzyme isn't bound, or the inhibitor isn't bound to the enzyme. So the E is free here, and it can bind either the inhibitor or the substrate. So you can imagine a situation where you have lots of an inhibitor present. So that makes this interaction more likely to occur. And so you have enzyme and inhibitor binding as the predominant. So it's like a chemical reaction that if you add inhibitor to this side, it's going to favor this state over here then. You remove the inhibitor, and it's going to start favoring this state with the free enzyme. So you can imagine that if you added enough substrate, so you keep adding the substrate here, you add substrate down here, then this reaction is going to be favored where the enzyme and the substrate is going to, the enzyme is going to bind the substrate, and then the substrate reacts and you have your product. So a key feature of competitive inhibition is that you can overcome it if you add more substrate. Allosteric inhibition is different on the other hand. So allosteric involves the inhibitor, that's this molecule over here shown in green, binding to an allosteric site. So that's allo means other, so it's a site other than the active site on the enzyme. So it binds to the allosteric site. This changes the shape of the molecule. So now you can see the active site has changed such that the substrate can no longer bind. And so you can imagine this situation here where instead of having the enzyme and the inhibitor competing, you either have the enzyme and the inhibitor bound, or the inhibitor isn't bound and you have the substrate. So I put the I on the other side here to indicate that it's binding somewhere else on the protein. So you can keep adding more and more substrate to this, but you're not going to get a reaction to occur because it's not competing for this site over here, but it's competing for the active site, which the active site has changed. So allosteric inhibition can't be overcome by adding more substrate, and that's kind of the key feature of that. Now we have allosteric inhibition, but you can also have activators binding to the allosteric site. And so in this case, the green molecule or the green shape is acting as a allosteric activator. It changes the shape of the molecule, so now that it's going to actually bind to the substrate better, so that the substrate can come in, you have your induced fit occur, and then the reaction actually occurs where you could break these two off and you'd have a new reaction. All right, so why enzymes are important is because they are very important for metabolism. So metabolism is just all the chemical reactions that are going on inside your body. And you have two types of metabolic reactions occurring. So the first is anabolic reactions. So anabolic, you can think of this as the anabolic steroids, where the reaction is building things up. So anabolism builds, anabolic steroids help you build muscle, so this is the building. So here, this is going to require the input of energy, so energy comes in, you have A plus B then being used to produce C. So you're building up a molecule from smaller molecules. Catabolism, on the other hand, is the breakdown of things. So here you have C on the reactant side, and you're breaking this down into A plus B, and it's also producing energy as a byproduct. Now, involved in metabolism are different biochemical pathways. So what a biochemical pathway is, so each of these different colored ovals represent an enzyme. And you have your substrate here, it can come in to the first enzyme, it reacts, and you have your product coming out. And this product then acts as the substrate for the next enzyme, and it can come out, and then this product acts as the substrate for the next enzyme, and then finally your final enzyme acts. And so you can just have this carried out. But metabolic reactions don't always flow linearly. Sometimes you have branching off points. And so that can occur here, for example, where this product here can actually go over, produce a different reactant, or it's the same reactant, but it's coming in and it's going to produce a different reaction so that you have a new product being formed. So instead of going over into the yellow, it went over to the purple enzyme and you had a different reaction occurring. Now, your cells and your overall metabolism want to be run efficiently. You don't want this reaction running all the time, and you don't want this reaction running all the time. Sometimes you'll want this reaction to occur, sometimes you want this reaction to occur. Usually this happens when you have a lot of this final product being formed. So if you have a lot of this final product being formed, it can actually act to help regulate it. And it could bind any one of these enzymes here and shut down the reaction in order for it to prevent it from going. To be the most efficient, it's going to want to bind the earliest enzyme in this reaction that's going to be specific to this specific pathway. So this enzyme right here wasn't specific because it could produce this product or this product. This enzyme wasn't specific because, again, it can produce either of these products. And then but if we get to this enzyme right here, it was specific. It didn't react over here with this enzyme. This product of this didn't react over here. So this would be the first specific enzyme to this biochemical reaction. And so this could act as an allosteric inhibitor for this, bind to it, change the shape, and now this is allosterically stopped. So when you have a lot of this being produced, this stops this, but you can still produce this if your cells need it. All right, I hope that was informative for you. You have a basic idea of how enzymes work in biochemical pathways and how they're going to be regulated. In class, we're going to be working on applying some of these methods and looking at getting a deeper understanding of this. So I hope you enjoy this video, and I'll see you in class.